Hello friends, welcome back to the journey of microbiology. Till now we discussed about in this chapter that's the general characteristics of microbes. It's a second unit in microbiology syllabus. In this particular unit we already discussed about how the microbes turns into the pathogens, and the first microbes, that is a bacteria, we already discussed in detail in the bacteriology. In today's lecture we will cover the two things. The first one is the bacteriophage, virus of bacteria, and second thing virology in details. Hope you all are clear with the previous topics. So today, see the lecture outline in the slide. In this lecture outline we already discussed about the structure and classification of microbes, morphological types, size and form of microbes or bacteria, their mortality, colonization, growth and nutrition of microbes. In today's lecture we will discuss first the bacteriophage. And then we will describe the virology, that's the study of virus in detail. So bacteriophage, as we know, it's a virus of bacteria which have the great, we can say, research importance, great medical importance. That's of the great medical importance. So in this lecture we will discuss the first bacteriophage. Bacteriophage is a virus of bacteria. It infect the bacterial cell. It infect the bacterial cells. So it's a virus of, the virus of bacteria. Okay. It affects the bacteria. Another thing about the bacteriophage is it's have great importance in the field of research. Important in the field of medical research. It's a tiny, we can say, the creature or organism which affect the bacteria. Suppose, see, this is one typical bacteria. Okay. Bacteria have their own their genetic materials called the nucleotide, and they have all other structures. See what happened. Bacteriophage it is a one typical bacteriophage. I will tell you in detail in subsequent lecture that's a bacteriophage, and it invade upon the cell wall of bacteria, and slowly what happened, they also have genetic material. Okay, they also have, I already told you in the previous lecture that every living creature possesses the genetic material, and there is no any exception about this sentence. Every living creatures contain the genetic material. So bacteriophage also have genetic material, and after the inversion it dissolve basically the cell wall of bacteria, and their genetic materials comes into the bacterial cellular environment. Okay, it affect the bacteria. Now, during the time of searching of the genetic material, now today's we all know that okay, the deoxyribonucleic acid, that is the DNA, is the genetic material present in most of the organism at least, and in some of cases like some of viruses, RNA also serve as the genetic material. The ribonucleic acid also serve as the genetic materials. So there is a long discussions, we can say the debate between the biologists that okay, what is the genetic material, what's the chemical composition of the genetic material. In the we can say the discovery of genetic material, this bacteriophage plays the crucial role. It plays the significant role. What happens? Scientists are able to radioactive their genetic material, radioactive. Okay, they radioactive it with the two things, the sulfur and phosphorus. Okay, and then they allow to invade the bacteria. So what happened, their genetic material is now identified with the radioactive substance. इनका जो genetic material है ये अब मिल चुका है this radioactive substance के कारण. It means if this genetic material will affect this virus, okay, their genetic material, it shows that the DNA is the genetic material present in every creature. DNA is the genetic materials present in the every creatures. Even though there are some viruses, there the RNA is present. Clear? That's the one clinical significance about the bacteriophage. Now, bacteriophage contain either the single standard RNA molecules as a genetic material, or they can contain the double standard DNA molecules as their genetic materials. Never both. Never both. It may be the single stranded RNA or it may be the double stranded RNA, but never both contain as a genetic material in viruses. Okay, in this bacteriophage. Now, come to their morphological appearance, how they look like. See, it's also a tiny organism, so it will visible only in the microscope. So I am going to draw the morphological diagram of bacteriophage. Bacteriophage have one head, the head portion. Okay, they have one head portion along with the tail. Along with the tail, they have one collar. They have one collar along with the tail. They have one collar, and in the head, the genetic material is present. In the head, the genetic material is present. Okay. See. Okay, I should draw it in a bigger way so it will be visible properly. See, I will going to identify the parts. This portion is a head portion. Okay, it is their genetic material. It might be either RNA or DNA, genetic material. Okay, now this portion is called collar of bacteriophage. Okay, then they have the sheath. Then they have the sheath. They have the sheath made up of the lipoproteins sheath. Okay, they have the tail sheath, and these two are the base plate, or we can say that pins, tail pins. It's a one typical structure of bacteriophage. That's the typical structure of bacteriophage. Now, the importance of the part, these parts help in the attachment, adherence on the bacteria, and this is the infected part. Okay, in the case of viruses, we will discuss in a few minutes that the genetic material is the infected part in the viruses. Okay, and it's also one type of viruses which is infect not the humans but the bacteria. Clear? Now we are going to discuss the one of the most fascinating and challenging branch in medical science, that's called the virology. So till now we discussed about the bacterial everything, the bacteriological everything except the staining procedure. So after describing all the microbes, I will deliver also lecture on the staining procedures. Okay, types of the staining and their uses, their applications. So for now we are going to discuss the virology. See, you might infected by some kind of viruses in your lifespan till now. You have the exposure of viruses and you commonly seen the sign and symptoms, you felt the sign and symptoms of the viruses. Okay, now study of virus is called virology. The study of virus is called the virology. Study of viruses. Study of viruses. Study of viruses is called the virology. There is no any clear-cut evolutionary relationships is shown till now by any evolutionary biologist. Okay, they don't have any kind of evolutionary history as compared to the other creatures like bacteria, other creatures. They don't have any proper evolutionary history. So we can write like they lack the evolutionary aspect. Okay, there are hypotheses, but they are not clear. They are just a hypothesis. They are not a clear concept. Okay. Another point about the viruses is it is smaller than bacteria. Their size is generally measured in the nanometer. Okay, whereas the bacterial size is measured in the millimeter, mm, but their size is measured in the nanometer. So they are smaller than bacteria. Okay, and under the classification we studied in the previous lecture, you might remember it correlate with now that we studied in the RH Whittaker classification system. They described every organism into the 5 category, but there is two exceptions. There's two exceptions. The first exception is viruses didn't get any place in Whittaker's classification system. Because why, I will tell you now. So it don't have any Whittaker's classification system, so it's a borderline between living and non-living. Between living and non-living. See, viruses exhibit some of the characters which look like a living organism character, but they also exhibit some of the character which shows that it's a non-living characters. So what are the characters? First one is a typical virus. A typical virus contain the universal thing, that's a genetic material. Okay, either RNA or DNA, never both, either RNA or DNA. Whereas about the other, we can say the cell constituents, they lack cell wall, they don't have any kind of we can say the ribosomes, any cellular machinery. They lack cellular machinery. That's why they are the one who evolve very faster. You seen in the case of coronaviruses how many strains, like in every, we can say, the place, the new kind of mutations arises. Why? Because viruses doesn't contain so many organelles, so many cellular structures, so they evolve quickly. They don't need to express so much gene to synthesize so much proteins. Okay, they generally contain simple things. If we talk about the viruses structures, viruses mainly contain their genetic materials, that's called the genome. Genome is basically the set of whole chromosomes, whole genetic material, that's called the genome. They first contain the genome. Second thing is the capsomer. For protecting those genes, viruses developed certain proteins. They express certain genes which result into the formation of one capping, that's called the capsomer. And that's the universal two constituents, only two constituents. See how like little things they create. Okay, two things. Then some of viruses, in the some of the viruses, they may contain the outer covering, that's called the envelope, for their protection, capsid protections. But the generally two things is present in the viruses. The first one is their genetic material, that's called the genome, and the second thing which present in the viruses is called the capsid, protein layer. So now they evolve faster. Now you know why they evolve faster. They evolve, or basically leave the evolution part, they mutate faster. They mutate faster rate, okay, as compared to any other creatures. They mutate at the faster rate. Now few more about the viruses. Their genetic material is either RNA, okay, or DNA. Genetic material is either RNA or DNA. They lack complexity, as we discussed. Complexity. They lack complexity. That's why they evolve faster at. Okay. Now, viruses size is in a nanometer. See, if we talk about the structure of virus, if we talked about the structure of viruses, virus generally contain the three things. If I draw the one typical diagram of virus, see, okay, the infected part, the red, this red marker showing the genetic part of the virus. Okay, you know, probably you know about it. There is one central dogma in molecular biology which states that the information is passed from DNA to RNA, and which result in the formation of protein. That's called the central dogma in molecular biology, given by the Watson Crick, a scientist, a microbiologist, who given that always whatever information is present in the DNA in the form of gene, they transfer to the RNA, and RNA is the one who expressed as the formation of protein. So what happened, this bacterial DNA, they form one RNA, that's a messenger RNA, and eventually after forming the RNA, it code, basically we can say it's code protein, it's code a protein layer. It's code a protein layer around the the viral DNA. Okay, and this coating is called capsid. This coating is called capsids, and it is a genetic material. It might be DNA or RNA. So we simply write it here, the genetic material. There are only two types of genetic material present in all living creatures. It may be DNA or it may be RNA. Okay, it's a genetic material. Now, like the other creatures, they are also able to adapt the harmful conditions. Okay, so what happened over a period of time, viruses develop one more kind of layer which may be present or not be present. Okay, that's called the envelope. The envelope is basically the lipoproteins layer derived from the host. So also they develop See, that's a envelope. That's a envelope. Okay, it develop. It not all in every bacteria, every viruses this envelope is present. No, it may be present or may not be present. So the viruses where the envelope is present, that virus is called the enveloped virus. That virus is called the enveloped virus, and the virus where this envelope is not present, they are simply called as necked virus, means khula hua, necked virus. Clear? Now, after discussing the structure of these viruses, we have to tell us about their growth and metabolism. So growth and metabolism. So before describing the growth and metabolism, see the slides, what information slide does. Virology. Viruses are autonomous infectious particles. Autonomous, mark the word, autonomous means self-driving. It's a self-driving infectious particles. They have no cellular structure. I already told, they don't carry so much complex in their life. So they are the one who mutate faster. They are mastered in the adaptation. So they have no cellular structure consisting of proteins and nucleic acid. Only the we can say their genetic part they only carry. They have no metabolic system. They don't have the digestive system, respiratory system, nervous system. No, they don't carry anything, but rather depend on the synthetic mechanism of living host cell. Once virus capture the host body, they take over their cellular mechanism and they take the nutrition from their we can say their cellular structures. Okay, so they have no metabolic system of their own, but rather depend upon the synthetic mechanism of living host cell, whereby the viruses exploit normal cellular metabolism by delivering their genetic information, that is nucleic acid, into the host cell. Clear? Now see some of the bacterial structure. First one is poliovirus. As you seen, and the things you keep in your back of your mind, that viruses not only affect the human body, it also affect the bacteria and plants and other animals also. Okay, and there is no any harmonious or the commensal relationship, or we can say the normal flora exists in the form of virus in our body. We initially born in this planet virus free. Okay, if we acquired virus, it will affect only us. It will no any beneficial. There is no any beneficial report till now coming from the study of viruses. So there are some structure, as you are seeing. The first structure is from the poliovirus. Second structure is adenovirus. Third structure is smallpox virus. Third and fourth structure is herpes virus, and the fifth structure is the influenza virus. As you are seeing, they are kept under the one bacterial cell structure. That's the Escherichia coli. That's the E. coli bacteria. And see, in one bacteria, this diagram is meant for the comparing the size between the bacteria and the viruses. As you are seeing, the E. coli structure, they are in the large, and inside that there are so many viruses, and now you can compare the size. Now, morphology and structure of virus. The pathogenicity of a virus depends on great variety of structural and functional characteristics. The pathogenicity of a virus depends on a great variety of structural and functional characteristics. I already told you that some of the viruses contain every genetic material, every cellular component, like they have the envelope, and they may not have the envelope. Okay, so this envelope and non-envelope structure of viruses also denote their pathogenicity, also express their pathogenicity. Some of the cases you've seen that okay, enveloped viruses are the more dominant to cause a disease, to cause the infections, whereas the unenveloped viruses, they are less potent, okay, they are less infectious to the living animals. Now additionally, some viruses contain envelope. Additionally, some viruses contain envelope. We already discussed. Viruses' simple structure and exposed genetic material contribute to their fast mutation rate, I already told, allowing the rapid adaptations and evolution in microbiology. Okay, now see one diagram, that's a non-enveloped virus and enveloped virus. As I told, in the case of enveloped virus, they are well covered by the one envelope, the lipid proteins envelope, and in the case of non-enveloped virus, they have only two things, the capsules and the nucleic acid. Now the first component of the viral structure, that's called the genome. Genome is basically the set of chromosomes. Genomes is the entire genetic material. They express the entire genetic material. So both the type of genetic material can be found in the viruses, but both cannot found in one viruses. Okay. Mostly in the nature, single stranded RNA and double stranded DNA is prevalent, but other form is also present with medical significance. Mostly the single stranded RNA and double stranded DNA virus is present, mostly, but there are some viruses which contain the single stranded DNA and double stranded RNA also, which have the medical importance, which have the medical significance. Now example of single stranded viruses is rabies viruses. You know, like when the dog bites, it is result into a disease called the rabies. Okay, so rabies virus, chikungunya, you probably heard about it. Chikungunya virus, Ebola virus, okay, measles virus, Hanta virus, that's come into the like recent discovered virus, Hanta virus, then HIV, human immunodeficiency virus, okay, then hepatitis C virus and dengue virus. These all are the single stranded RNA virus, whereas the double stranded DNA virus are adenovirus, okay, papilloma virus, Vacilla zoster virus, cytomegalovirus, herpes simplex virus and hepatitis B virus. You have to mug up all these name of the virus because they may be asked in the exam in the flowing which are the single stranded virus, which are the double stranded virus. So it's in tip of your finger. It will be a tip of your finger so that you will not confuse. Now come to the capsid, the covering of virus. A protein coating structure capsid is designed to protect the genome. I told you no, genome expressed to form the one structure for their protection point of view, that's called the capsid. For most viruses family, capsid shows two type of geometrical configurations. The one is called helical, another one is called isohedral. Okay, helical is basically the rod shaped or coiled shaped, whereas isohedral is a spherical or symmetrical in shape. The capsid is constructed of multiple copies of polypeptides required less number of genes to form capsid. Capsid is made up of capsomers. Because the nucleic acid of a virus is surrounded by the capsids, it protects from the environmental damage. It's protect from the environment. And now see the two types of diagram of the capsid. The first diagram, as you are seeing, it's a round. It's like a helical in shape. It's a spherical in shape. That's the helical configurations, and another one is the isohedral. Okay, now I have to draw it so that you will understand it in a proper way. See, we discussed about the genome. Okay, genome is basically their genetic material, okay, by which they infect the creatures, they infect the living creature. That's the genome, and genome basically represent the total chromosomal pattern. Okay, total chromosomal pattern. That's the infectious part of the virus. It is the infectious part of the virus. Now come to their capsid, the protection part. I told you viruses express certain proteins which forms the one protective covering for their genome, for their genetic material. Okay, they form the one covering for their genetic material. Now about the capsid, there are two types of capsid. Geometrical point of view, geometrical structure, there are two types. The first is the helical, and another one is isohedral geometry. See, helical structure is nothing but like, okay, see, it is a helical structure. It's coiled structure basically. It is a coiled structure. It is a coiled structure. And inside this coiled structure, the viral genetic material is present. That's the one type of, it's a helical structure. Okay, helical configuration. Now in the isohedral configurations, they are like, okay, this, okay, and in this little space the viral genetic material is present. Okay, so it's basically the type of isohedral configuration. It's a isohedral configurations. Clear with this? Now the unit of these capsid is called capsomer. It will ask surely in the MCQ, capsomer. Okay, capsomer is basically one unit of capsid. It's a one unit of proteins. Clear? Now about the capsid, two more things. It's already told, no, it's composed by the expression of genome. It's composed by the expression of genome. Now we will discuss the another part of the virus, that's called the envelope. I told you envelope may be present or may not be present in the viruses. It may be present or it may not be present in the viruses. If it is present, it's called the enveloped virus. If it's not present, it's called the unenveloped virus. Envelope. Okay, envelope, it may be present or it may not be present in the viruses. It is derived basically from the host cell membrane. See, if I tell you briefly how viruses affect one cell, see, this is one typical virus. One typical virus, okay, covered by the good capsid, and this single unit is called, very good, capsomers. It's a capsomer. Okay. Now in some of viruses, the envelope will present, and in some of the viruses it may not be present. Okay, so this virus, in fact, suppose this is one cell, we can take the example our respiratory cell. Okay, respiratory cell. Now it attacked here. When it attacked, their genetic material comes here. Okay, till now this portion is only present, this envelope is not present, but when it enters into our cellular environment, where we have the proper bound protected nucleus, we have protector covered by the nucleic covering, we have the proper nucleus where our genetic material is preserved. This viral genetic materials first comes into this cytoplasmic space, our cytoplasmic space, and we have so many organelles. Okay, we have so many organelles. We also have lysosomes, which carries the destructive enzymes. So what happened if this virus comes here? Okay, they are exposed by the lysosomes, they are captured by the lysosomes, and they will discard. If they are fortunate enough, they are blessed, they escape from the lysosomes. Okay, and then either they start affecting the cytoplasm or they directly capture our nucleus. In the case of HIV virus, that's a AIDS, in the case of AIDS disease, the virus genetic material is RNA. Okay, and they start destroying our T or B helper cells, T and helper cells. So person becomes the immunocompromised. So it's a basic transmission of viral genetic material. Now, after discussing the structure of virus, we will discuss the virus replications. See, they don't need nutrition. So it's a viral replication, the one-step growth curve. Okay. In the case of virus, they don't need the nutrition. Okay. They only need to copy their genetic materials. Okay. So for copying of the genetic materials, that process is called the replication. Very good process is called the replication. So we will now discuss the viral replication. Okay. Replications. So first read the slide, then we will discuss it in detail. The one-step growth curve begins with two things: the ellipse period and exponential growth. In the ellipse period, viruses infection particles affect the host cell. During this period, active synthesis of virus component is occurring. The ellipse period of most human viruses falls within a range of 1 to 20 hours. The number of progeny viruses produced within an infected cell increases exponentially for a period of time, then reaches a plateau, after which no further increment. They may be formed 8 to 72 hours or longer with yield of 100 to 1000 virions per cell. See, as you remembered, in our previous lectures, we discussed the bacterial kinetic curve, the bacterial growth curve, in which we plotted the bacteria against the time, the bacterial cell numbers against the time, and we seen bacteria grow very fast. Okay, now it's point to discuss the virus cycle. See, in the case of viral replication, they have one single curve. single curve. Okay, this curve is denoted by the two types of growth pattern. The first pattern is ellipsed, second one is exponential. Okay, now first I will draw the viruses exhibit two kind of growth pattern. The first one is called the ellipse. Okay, here it's a C, ellipse. Okay, ellipsed. First one is ellipse pattern growth and second one is exponential growth. What happened when viruses enters into the cell? Okay, they are exposed to the host cell's membrane. Host cell membrane, and their genetic material is inclined or start embedding on the cell membrane. During that period, there is no any growth in viral genetic material. Okay, so I will draw the diagram. I will first draw the diagram and then explain. Okay, now in the case of virus, the diagram is versus infectious viruses per cell versus the hour. It's the hour means time, and infection proceed, or we can say affect the cell. Okay, it's the diagram. Now initially what happened, bacterial growth decline, then take one period and then their growth. That's one single step. In the case of bacteria, we seen first they have the log phase, then they have the lag phase, then they have the exponential phase, and then they have the die phase. But in the case of viruses, generally what happened, it first infect the cell. Viruses first infect the cells. During the infection, during the adhesion, basically the adsorptions, viruses not suddenly start increasing their number, genetic material. No, they first try to, we can say, like compensate, try to compromise with the host cells. Okay, they are very chalak, they are very clever. They first try to make one proper relationship with the host cell. Okay, and then what, after the establishing one relationship, they hijack their cellular mechanism, and now see how fast they start to infect the cells, So that's called the one single step curve. Now mostly in the ellipse period, in this period, if I will tell you, see, if this is a virus, okay, and this is one typical cell, it start. So after infecting the cell, okay, they comes here, their genetic materials comes here, okay, and they loses the parts. These all parts is discarded, all these parts are discarded, only the genetic materials come into the cell. Okay, now here no any growth. See, this is the period, no any growth. Okay, no any growth. They start synthesizing their parts, like these parts. They start synthesizing the capsid. Some of the cases, if the virus is enveloped virus, then they start to synthesizing the covering. Okay, and either they will strict with the mRNA. If it is RNA virus, they will strict with our own mRNA, or either they attack our well protected genetic material, that's the DNA. Okay, if it's RNA virus, it will only present in our cytoplasmic space. If it is a DNA virus, it will present in our nucleus, it will affect our nucleus. So during this period, hijacking period, there is no any growth in the virus. Okay, they see, virus doesn't need growth, no, they don't need growth, they need infections. Okay, ये चाहते हैं इनका ग्रोथ, इनका जो रेप्लिकेशन है वो इस चीज़ के बेसिस पे जज किया जा सकता है, डिटरमाइन किया जा सकता है कि कितना सेल को इन्फेक्ट किया, कितना लिविंग सेल्स को इन्फेक्ट किया. दैट्स व्हाई वी आर प्लॉटिंग द ग्राफ बिटवीन द infection proceed, infected affect the cells versus the hours. In the case of bacteria, we plotted the graph between the bacterial cell number along with the time. So here suddenly we got the viral exposure and we become sick. No, it's not happened. It takes some certain time. It's also explained why our, like we can, the successive exposure of viruses doesn't lead sudden sign and symptoms. This period explain also this thing. Okay, now in the ellipse period, two things happen. First one is active synthesis of all those required things. Okay, and it will take, there's one time period, it will take the time 1 to 20 hours. It will take 1 to 20 hours. Okay, it will take 1 to 20 hours. Clear? Now about the exponential phase, about this phase. The exponential phase, in this phase the actual infection proceed. In this phase, I should change the marker. In this phase, the actual infection is proceed. Now, virus is well prepared. They are well groomed with their necessary things. They hijack the machinery. Now, their second motive is what? Their second motive is to make viral progeny, to make the viral species more and more. So in this period, this is the period where the actual viral, we can say, the progeny is produced. Viral progeny is produced. Clear? Viral progeny is produced. Okay, it takes time, this to 72 hours. 72 hours. Okay, 72 hours. Now we will now discuss the steps in viral replications. Okay, see the board, steps in the viral replication. This is the one pattern. Okay, that's a graph. Okay, and this graph explains that the viral replications occurs in the one single curved steps. Okay, and these steps, there is two types of growth pattern is shown. The first pattern is called the ellipse pattern. The second one is called the exponential growth. In the ellipse pattern, we seen viruses only attack the host cell. Okay, and then they try to compensate, they try to make the relationships. If they survive, then they start active synthesis, and in the exponential phase, the actual replication of their progeny is started. Now we will see how viruses replicate. Till now we discussed roughly, but now we will go with in the sequence how replications actually happen. Okay, replication. Replication is a process by which genetic material is copied. It's called the replication. Viral replications start with the first step, that's called the adsorption. First step. Adsorption. First step is adsorptions. In this phase, two things happen. In this phase, two main marked things happen. The first thing is attachment. attachment, and second thing is receptor. Second thing is receptor attachment. As you know, our cells carries in their cell membranes different kind of receptors, okay? And one thing more, each receptor is specific for different of viruses, okay? Not every receptor allow every type of viruses to come inside the cell, okay? Every receptor is specific towards the specific type of viruses, okay? So what happened in this adsorption period? Adsorption is basically the phenomenon, okay, the phenomenon by which one substance attached to the another substance. That's called the simple adsorptions. Whereas in the concern of the replication adsorption, okay, viral adsorptions, it happens into the two things. First one is attachment. Viruses attach the host cell and then they attach their, see, if this is one typical virus, okay, and that's our eukaryotic cell, okay, and this is our cell membrane. Cell membrane is basically, we know it's a phospholipid bilayer. It's made up of the phospholipid bilayer, okay. So on the surface of cell membrane there are receptors, okay. There are 7 parts receptors, G proteins coupled receptors, single receptors, double receptors, okay. We have so many kind of receptors. So there is a receptor. So virus first comes and attach, and then they attach with the receptors. They won't allow directly. Our this cell membrane is meant for protecting. They are the selectively permeable. No, they don't allow everything to come inside. So they have the receptors, and this virus comes into the receptors, and after the receptor there is one signal is generated and virus genetic materials come inside, okay. Now what is the clinical importance of understanding this step? The clinical importance of understanding this step is that by understanding the mechanism how virus is attached to our cells, we can say the doctors are able, okay, the pharmacists are able, the chemists are able to create certain antiviral drugs which disrupt here, okay, which disrupt here. They just block entry. If by some exogenous substance, that's called the drug, okay, this things is blocked, then if our one cell is affected, okay, virus successfully enters on that cell, okay, and they replicate, they make the progeny, but they don't affect the another cells. So there are some antiviral drugs which work here. Antiviral drug, okay, which block these receptors and they won't allow bacterial genetic materials to come inside, okay. Now after discussing the adsorptions, we will discuss the another step in the replication of viral genomes, that's called the penetration. Penetrations literally means embedding inside. Penetration, okay. The second step is, okay, we will write one type here, the steps. First steps we discussed adsorptions. Now the second step is penetration. Second step is penetration. Second step is penetration. In the penetration, the well formed virions, okay, well formed the virus particles come inside the cellular machinery, Till now in adsorption we seen, okay, first is attached, then it's attached on the receptors. Now it's point to come inside the actual cellular environment. See, this one eukaryotic cell, okay, and after attaching with the cellular receptors, the viral genetic materials transferred into the cellular environment. This process is called the penetrations. penetrations. Now viral genetic materials enters inside the cells. Okay, they are exposed to our own cellular environment, and you know, we are also mastered in art these little things. They are thinking, okay, we are wise, but they don't know humans are too much wise than them. They already discovered antiviral drugs also. Okay, and sometimes they are challenging. In the case of HIV, they are challenging. There is no any proper we can say the treatment. Okay, we have only the symptomatical management. So they enters, but our cells are also masters in the art of protecting us. So they have so many things. Okay, they have lysosomes. Okay, and lysosomes is called death pocket. Okay, it's a cell death pocket. We have the lysosomes. Okay, so what happened? This is first exposed to the lysosomes. If lysosomes are like potential enough to sustain the growth of virus, to discard the virus, we don't have any signs and symptoms of viral disease. But due to certain, we can say the immune deficiency, or we can say certain enzyme deficiency, they don't destroy. अगर ये हार जाते हैं, then they will able to start their replication process further. Okay, replication process further. So in this step, two things happened: receptor mediated endocytosis. Don't confuse with these terms. Endocytosis means simply they allow to come inside. Endocytosis. Exocytosis means outside. Endocytosis means engulfing anything inside, and exocytosis means making things outside. Okay, so basically they allow to come. That's called the receptor. This is a receptor mediated endocytosis. Okay, and second thing is membrane fusion. Clear? Membrane fusion means we have no membrane bound organelles, so they are fused with those membrane bound organelles. Okay, now the third step is uncoating. So before describing the uncoating, see the slides. The adsorptions, we discussed already the adsorptions: attachment site on the viral surface, and the host cell receptor molecules. In the information about the three-dimensional structure of virus binding site being used to design the antiviral drugs, as I told, that specifically interact with these sites and blocking the viral adsorptions. Now second step, the penetrations. Two things happens. Penetration means passing the virions from the surface of the cell across the cell membranes into the cytoplasmic space. Okay, it occurs in the two methods. The first one is called the receptor mediated endocytosis, or second is the membrane fusion. Now see the diagram. There are four diagrams. Okay, in the first diagram you are seeing that the viruses bind to our receptor cells, host receptors, or invaginations of the membranes started. They welcome the virus particle inside, and after welcoming, they fuse with our vesicles. And they protect they covered their genetic materials by the endocytotic vesicles. Okay, and in one case you are seeing the viruses don't bind directly. Okay, they don't bind directly because drugs are already came and they just cover those parts. Now the third step in viral replication is uncoating. Uncoating basically refers to the stepwise process of disassembling of the virions that enables the expression of the viral genes that carry out replication. Okay, it's basically the stepwise process where the virions becomes expressible. I will tell you what is a virion. Okay, for enveloped virus, the penetration process itself is the first step in uncoating. In the enveloped virus, the penetration process itself is the first step of uncoating. The loss of one or more structural component of the virion during uncoating predictably leads to the loss of the ability of that particles to infect other cells, which is the basis for the ellipse period of the growth curve. It is during this phase in the replication cycle that viral gene expressions begin. Now, see here, the third step in the viral replication is uncoating. Uncoating. The third step in viral replication is the uncoating. Uncoating. What happened till now? We've seen, okay, there is one virus, okay, which comes, viral genetic materials come inside, okay. Now, they come inside, they either attack the nucleus or the cytoplasmic organelles, okay. Now, after the attacking this portion, the entire portion is called virion, okay. It don't have any kind of envelope, it don't have any kind of capsid also, so it's just a called virion. We can say the exposed viral genetic material is called virion. The infectious particles of a virus is called virion. Clear? Okay. Now, these genetic materials now need to replicate. Till now we are seeing, okay, enters, how uncoatings happen. Now it's time to understand how this is replicating. This is the step where the viral genetic material start to replicate. Clear? That's called the uncoating process, uncoating step. In the case of enveloped virus, penetration in the penetration process itself, the uncovering process start. Enveloped virus has the outer covering. So in the penetration process, already when the receptors attached start, okay, receptors start and endocytosis, receptor mediated endocytosis occurring. On that very moment, uncoating started in the case of enveloped virus. Whereas in the case of non-enveloped viruses, this thing happens, uncoating happens in this on particular process, and it is the period where the virions, the actual infectious particles, start to replicate means increasing their number. Replication means 1 to 2, 2 to 3, 3 to 4. The genetic materials progeny start replicating, increasing their number. Now, that's the third step. Now the fourth step is mode of replication. Okay, the fourth step is mode of replication. Mode of replication. We can also say the mechanism of replication. Mode of replication. See, we have two kind of viruses. The first one is DNA viruses, and second we have RNA viruses. Okay, simple. I already told. No, DNA viruses start replicating, expressing their genes, okay, and they result into formation of mRNA, messenger RNA, okay, and now messenger RNA turns to produce the proteins. That's one way. Clear? After entering inside the cell, it starts replicating. There is a mode, okay, there is a mechanism how. In the case of DNA viruses, they form the mRNA, they express certain genes which result in the formation of mRNA, and mRNA code for the particular kind of proteins. And virus need how many kind of proteins? Two kind of proteins, no? One kind of proteins. For what purpose? For purpose of capsomer formations, to protecting their genetic materials and they start again affecting another cells. because of that covering, viral proteins are protected from the antivirus drug also, and from the external any lysozymatic enzymes, okay, any other enzymes also. Now in the RNA, the reverse transcription process occurs. In the RNA, what happens? They code for the proteins. That's called the reverse transcription. Reverse transcription. See, from DNA to RNA, it's a transcription, and from the mRNA to protein, it's a translation. It's a translation. So you see the diagram, the mechanism of DNA virus genome replication. Mechanism of DNA virus genome replication. See, in this diagram, you are able to see that the parental DNA start to forming the mRNA. I told you mRNA, and these mRNA result into the formation of proteins, and that proteins result to formation of a virion. That's a virus. Clear? Now see the another mechanism for the RNA genome replications. In the RNA genome replication, the single stranded RNA, they result into the mRNA, okay, formation of mRNA, and after formation of mRNA, they result into the formation of proteins, and after proteins formations, the virus progeny is formed. Virus protein progeny is formed. Okay, that's a capsid formation and genetic material. Okay, the virus capsid formation and genetic materials. That's called the mode of replication. That's called the mode of replication. That's called the mode of replication. Mode of replication. Now the last step, mode of replication. Mode of replications. Mode of replication. Now the second last step is assembly of release of progeny virus. Assembly. Okay, assembly of release of viral progeny. It's a E number. Assemblance of viral progeny. Now, assemblance of viral progeny denotes that after affecting successfully the one cell, okay, and making their replica, making their, we can say, the virions, various numbers of virions, they need to affect the another cell also. They need to affect the another cell also. Okay. So that's the step is called the assemblance of viral progeny. That's called the assemblance of release of progeny virus. Okay. Now see the slide. Assembly of nucleocapsid generally take place in the host cell compartment. Okay, host cell compartment where the viral nucleic acid replications occur. In the cytoplasm of most of the RNA viruses, and in the nucleus of most of the DNA viruses. See, either they will take the charge of nucleus, or virus either take the charge of the cytoplasm. That's only the two possibility, nucleus or cytoplasm. Either they assemble, वो इकट्ठा होते हैं या तो nucleus में, I already told you, or either they are assemble in the cytoplasm. Clear? Now, effect of viral infection on the host cell. That's the last topic for today. Effect of viral infection on the host cell. Effect of viral progeny on host cell. We can say here effects on simply the host effect of host. Okay, so see the diagram. When this virion successfully enters into the host, that period is called the absorptic viral phase. Then they protect viral infections, okay, into the progeny virus released, and they take the host genome charge and viral infections resulting in host cell death and production of progeny. See, I will draw one diagram and show what is the meaning of this replication, this effect of, see, this is one big eukaryotic cell, okay, big eukaryotic cells. I am concluding today's lecture, okay, and here the genetic material, okay. Our cell membrane, as you know, it's a double membrane layer made up of the phospholipids contain certain receptors, certain channels, we call it ion channels, and many more things, okay. Now in the first step, adsorption, the first step, as you are seeing, it's the adsorption. In these steps, what happened? The virus comes into the human body, okay. Now in the penetration process, what happened? This viral genetic materials comes along with their covering, along with their capsid, they come, okay. Then in the penetration process, they start to come inside, the genetic material start to come inside, okay. Then these coverings start to disrupt, and only viral genetic material is exposed, okay. In the mode of replication process, if it is a DNA virus, okay, then it translates into the mRNA, and mRNA result into the protein formation, okay. In the case of RNA, the reverse transcription process occurs, okay. Now mode of replication done, and mode of assemblance means these viruses either stick with the cytoplasm or they takes our own nucleic we can say our nucleus and start replication, clear. Now effect of cell is what happened. If they attack our nucleus, then they make so much progeny virus particles, that's called the virions, okay, and these virions is eventually released for the another cell to effect there, clear. Now, so for concluding today's lectures, we discussed in details about the bacteriophage, a virus of bacteria, okay. After that, we discussed about the virology. In virology, we discussed about the general characteristics of viruses along with their mechanism, how these viruses affect the cells, and cells becomes our body becomes we can say the ill, okay. So thank you, hope you enjoyed this video. Bye.